Welcome to Prospective Doctor's MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctor's MCAT Basics with your host, Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. This podcast covers fat and protein metabolism. I'll talk first about fat catabolism or breakdown, and this is going to include lipolysis and beta oxidation. Then I'll get into fatty acid synthesis, which is basically the reverse of beta oxidation with a few caveats. Next, I'll talk about protein breakdown, which is going to include deamination in glucogenic versus ketogenic amino acids. And then lastly, I'll touch briefly on protein anabolism or synthesis. And in that segment, I'll talk about dietary protein absorption and its overlap with central dogma. And this material is going to show up on 2 of the 4 MCAT sections. That's going to be the Biochem and Chem Phys sections. So I hope you find this podcast helpful in your studies, and good luck as you get ready for the MCAT. So I want to start with fatty acid metabolism, and I'll talk about catabolism or breakdown of fatty acids and anabolism or buildup of fatty acids separately. And I'll start with fat catabolism, which again is just the breakdown of fats to produce energy. And I want to start with a review on fat absorption in the lymphatic system. So when we eat fats, they travel from our mouth, down the esophagus, into the stomach, into the small intestine. And the digestion of fats begins in the mouth with salivary lipase. And then in the stomach, short-chain fatty acids, which are fats with less than 6 carbons, are absorbed via just diffusion. And short-chain fatty acids are things like propionate and butyrate, which you might be familiar with. Now, the longer fats don't get absorbed until they get into the small intestine. And further, these fats are not soluble in the watery solution that enters the small intestine called chyme. So essentially in the small intestine, they are emulsified, further broken down, and absorbed. So there's 3 big steps in terms of fatty acid absorption that occur in the small intestine. And what I hope you're kind of thinking right now to yourself is, what are these fats? Are they triglycerides? Are they free fatty acids? What types of fats are we getting from our diet? And I'll say that dietary fats are mostly triglycerides, which have a glycerol backbone and 3 fatty acid tails. In addition, we have some cholesterol that we eat too, although most cholesterol is actually synthesized in the body, something that most people don't know. Okay, let me take a step back real quickly. So again, we're talking about dietary triglycerides. So say you're eating something with olive oil, and olive oil is full of triglycerides. So these triglycerides start to first be broken down by salivary lipase in the mouth. Then they make their way into the stomach and are kind of squirted into the small intestine. And in the small intestine, triglycerides enter as these kind of big fat globules. And the reason they do this is because they are not soluble in the watery chyme solution that's in the small intestine, as I said earlier. So in the next step, these fats are solubilized by bile. And again, bile is secreted by the pancreas and it acts to emulsify fats, which emulsify is just a term meaning that it disperses the fat into these smaller droplets throughout the chyme in the small intestine. At the same time as these fats are being solubilized by bile, pancreatic lipase and colipase start to break down these triglycerides into 2 free fatty acids and a monoglyceride. Essentially, these enzymes are plucking 2 of the fatty acid tails off the triglyceride. It's like ripping 2 legs off a 3-legged insect. And if you're thinking there's no such thing as a 3-legged insect, you are wrong. There's actually an insect called Anabolism tristis, which sounds like a Harry Potter spell, but anyways, that exists. So again, these enzymes, pancreatic lipase and colipase, start to break down the fats that have been solubilized by the bile. And if you're wondering if there's a connection between the two, like do fats need to be solubilized in order for these enzymes to come and break them down? The answer is kind of. So these enzymes can actually bind to the bile salt, which actually helps them access these fats. So that's one factor, as well as when these fats are emulsified, their surface area is increased by a whole hell of a lot. And that also helps these enzymes kind of access those fats and continue to break them down. So pancreatic lipase and colipase don't necessarily need the fats to be solubilized to break them down, but this emulsification or solubilization by bile really does help them be more efficient in breaking down the fats. So in this process, triglycerides are broken down into monoglycerides and 2 fatty acids. And what happens is they retain their association to bile salts and complex with other lipids, then form micelles. And these are even smaller droplets of fat. So essentially, we go from these big fat globules to smaller emulsified droplets to even smaller droplets called micelles. And at this point, the micelles transport the free fatty acids and the monoglycerides to the enterocytes in the distal portion or end portion of the jejunum and the ileum for absorption. Now, once these monoglycerides and fatty acids make their way into the enterocyte in these micelles, they are resynthesized into triglyceride and packaged into what are called chylomicrons, along with cholesterol, fat-soluble vitamins, and phospholipids. And this is a little bit counterintuitive because obviously these enzymes in the mouth, as well as the small intestine, have worked really hard to actually break down these triglycerides into monoglyceride and 2 fatty acids. So why are we reassembling them into these triglycerides? And the answer to that is that I don't know, but that's exactly what happens. And then those triglycerides are packaged into these chylomicron particles. After that, chylomicrons are transported via the lymphatic system to the liver, to adipose tissue, to muscle, where they can transfer their triglycerides or other fat molecules to cells. In addition, these chylomicrons can transfer fat molecules to other carrier molecules like LDL or HDL, which are lipoproteins. And then these particles, specifically LDL, can actually deliver fats and cholesterol to the cells in the body. Okay, so to kind of summarize this fat absorption process, when we eat fats, which are mostly triglycerides, they are broken down in the mouth and also in the small intestine into monoglyceride and free fatty acids. These are eventually incorporated into very small particles called micelles that are able to be absorbed into enterocytes, which are just cells in the small intestine. Once inside these cells, the monoglycerides and free fatty acids are reassembled into triglycerides and combined with other fats to make chylomicrons. These chylomicrons then enter the lymphatic system, and eventually these triglycerides and other fats are distributed to cells throughout the body. Okay, so that's how fat makes it to cells. Now, once fat is in the cell, a few things can happen. It can be stored for later use, or it can be oxidized right away to make ATP, the energy currency of the cell. Let's talk about that first one first. So fat is mostly stored in cells called adipocytes in the form of triglycerides. And hormones like glucagon, which is released when glucose is gone, and epinephrine can induce lipolysis in the adipocytes. And all that's happening during lipolysis is that these triglycerides that are stored in the adipocytes are broken down into free fatty acids and glycerol and then released into the blood. In other words, this goes a step further than a lot of the enzymes that are in the digestive tract that break down fats, right? Because this basically rips off all the legs of the 3-legged insect. It breaks down these triglycerides into glycerol and 3 fatty acids. And with that said, I will say that glycerol is a blood-soluble molecule. So when it's released into the blood, it just travels in the bloodstream and is used for different things. On the other hand, these long-chain fatty acids are very hydrophobic, and therefore they're not blood-soluble. They need a carrier in the blood. And their carrier is a protein called albumin. And this is a protein that carries a lot of different things. It's a very common protein, but it has 6 binding sites for free fatty acids. And so once these fatty acids are bound to the albumin, they can travel around in the blood and eventually be delivered to different cells. And once these different molecules are in the cell, they can be used for different things. For example, free fatty acids can undergo beta oxidation, and glycerol enters the glycolysis pathway, which can also produce ATP. So overall, lipolysis is a reaction that breaks down triglycerides into glycerol and three free fatty acids, and it occurs in adipocytes that essentially store triglycerides in these big old droplets in their cytoplasm. Okay, with that said, now let me get into beta oxidation, which was the second thing I mentioned earlier as to what happens to fats once they get inside the cell. And let me just back up a sec to fully explain this. Reverse. Dietary fats, which again are mostly triglycerides, are transported in chylomicrons and LDL particles to cells throughout the body. And once these triglycerides are dropped off to the cell, they are converted to glycerol in three free fatty acids by enzymes in the cell. This is the lipolysis reaction that I talked about earlier that's going on in adipocytes. It can also happen in cells throughout the body that need to break down fats and start to oxidize them for energy. So this is all to say that fatty acids that are oxidized in our cells can come either from adipocytes in times of needed energy, say when glucose is low, or they can come directly from our diets. All right, so now let's get into the beta oxidation pathway. So beta oxidation starts in the cytoplasm of a cell. A fatty acid molecule must be activated by adding a CoA group to it. So when a CoA group is added to fatty acid, it becomes acyl-CoA. And the enzyme that does this is called acyl-CoA synthetase. And this process of activating the fatty acid requires ATP. All right, so once this fatty acid is activated, it's ready to be oxidized in the mitochondria. But it's not very simple for the fatty acid to get inside the mitochondria into the mitochondrial matrix. It relies on what's called the carnitine shuttle to actually get inside the mitochondria so that beta oxidation can occur. And you could think about this a little bit like a group of friends that gets all dressed up to go out on a Friday night. You know, they make their way to the club, they're all ready to go, but when they get there, they can't get in, right? Their name's not on the list. And so how do they get in now? Well, let's say that they know some famous person that's at that party. That person can come and help get them in the party. And that's exactly what the carnitine shuttle does. The carnitine shuttle begins with a mitochondrial membrane protein called carnitine palmitoyltransferase 1, or CPT1. And this replaces the CoA group that has activated the fatty acid with a carnitine group. Now, this fatty acid plus carnitine, called acylcarnitine, is able to pass through the outer mitochondrial membrane. And now we're halfway to the mitochondrial matrix where beta oxidation occurs. In order to get into the mitochondrial matrix, another membrane protein, this time on the inner mitochondrial membrane, called carnitine acylcarnitine translocase, shuttles the acylcarnitine across the inner mitochondrial membrane and into the mitochondrial matrix. Now the fatty acid has entered the party. Now, once inside the mitochondrial matrix, this acylcarnitine is converted back to acyl-CoA, which is the long-chain fatty acid just connected to a CoA group. It's that activated long-chain fatty acid. And I will mention that the activity of this carnitine acylcarnitine translocase relies on carnitine, right? So in the mitochondrial matrix, carnitine is removed from this long-chain fatty acid and now is this single molecule. And so what this protein does that sits inside the inner mitochondrial membrane is it moves a carnitine out of the mitochondrial matrix while at the same time moving an acylcarnitine into the mitochondrial matrix. Now, once this fatty acid is inside the mitochondrial matrix, beta oxidation can happen. And from a conceptual standpoint, beta oxidation is really not that complex. Basically, the long-chain fatty acid CoA molecule, again called acyl-CoA, is cut into a series of 2 carbon molecules. From there, a CoA group is added to each of these 2 carbon units to create acetyl-CoA molecules. And by the way, acetyl-CoA is not to be confused with acyl-CoA. Acetyl-CoA is a 2 carbon unit with a CoA attached to it, whereas acyl-CoA is a fatty acid with a CoA attached to it. So just keep that in mind. And if you remember, acetyl-CoA can feed directly into the Krebs cycle and can eventually go on to produce a lot of ATP. So at a conceptual level, what beta oxidation is doing is producing a bunch of acetyl-CoA from a fatty acid that feeds into the Krebs cycle and is used to produce ATP. With that said, I don't think it's all that necessary to know beta oxidation in great detail, but there's a few things I would know. That being the basic reaction, the amount of energy or ATP that is produced per long chain fatty acid, and a few weird cases like what happens when an odd chain fatty acid is fed into this beta oxidation pathway. And what about unsaturated and saturated fatty acids? Do those do anything differently in beta oxidation? So I'll go through each of these. The basic reaction. Each round of beta oxidation involves chopping 2 carbon units off the main fatty acid chain to produce a molecule of acetyl-CoA. And this reaction includes 4 main steps. In order, they are dehydrogenation, hydration, dehydrogenation, and cleavage. So essentially, those 4 steps are repeated over and over to chop 2 carbon units off this long chain fatty acid. Now, we want to give a little bit of an example here. So take palmitic acid. This is an unsaturated fatty acid that is 16 carbons long. That means that 8 acetyl-CoAs can be produced from it. Now, what you should understand is how many rounds of beta oxidation it takes in order to chop this 16 carbon fatty acid into 8 acetyl-CoAs. The answer is that it only takes 7 rounds of beta oxidation. If this doesn't make sense, think about cutting a piece of paper into 8 pieces. How many cuts do you need to actually make to do that? You only need 7 cuts, right? Because the 7th and last cut will actually yield 2 pieces of paper. And it's the same thing in beta oxidation. The last round of beta oxidation will actually produce 2 acetyl-CoA molecules. And so that is extremely important to keep in mind as we talk about the energy yields from this process. Energy yield. Earlier, I talked about how it takes 2 ATP to activate a fatty acid. So just kind of keep that in the back of your head as we talk through the energy yield produced by beta oxidation. Each round of beta oxidation produces 1 NADH, 1 FADH2, and 1 acetyl-CoA. But of course, remember the last round of beta oxidation produces 2 acetyl-CoA. So the beta oxidation of palmitic acid, which has 7 rounds of beta oxidation, yields 7 NADHs, 7 FADH2s, and 8 acetyl-CoA. NADH is worth 2.5 ATP, FADH2 is worth 1.5 ATP, and acetyl-CoA is worth 12 ATP. So a single molecule of palmitic acid will produce a whopping 122 ATP. And of course, don't forget to subtract the 2 it takes to actually activate the fatty acid. And this is much more energy than a molecule of glucose produces. Think about it. Glucose only produces somewhere between 30 and 32 ATP when it's been fully oxidized. As a result, fats per gram contain much more energy than carbohydrates. Weird cases. I want to start by talking about saturated versus unsaturated fatty acids. So saturated fatty acids are ones that are saturated in hydrogen and therefore don't have double bonds. We've already talked about the beta oxidation of these when I talked about palmitic acid. Unsaturated fatty acids are fatty acids that are not saturated in hydrogen and have double bonds. Now, these are handled a little bit differently than saturated fatty acids when it comes to beta oxidation. In short, fatty acids that have an even number of double bonds require 2 enzymes, an isomerase and a reductase, to convert the fatty acid into one that can actually enter beta oxidation. On the other hand, fatty acids that have an odd number of double bonds require only 1 enzyme, an isomerase, to convert it to a fatty acid that can enter beta oxidation. In addition, for these unsaturated fatty acids, you're not making as many of the high energy carrying molecules. So in the instance of a single double bond in an unsaturated fatty acid, you lose one of the FADHs. And then if you have 2 double bonds, you're losing an FADH2 and an NADH. So the takeaway is that you're not making as much energy from the beta oxidation of an unsaturated fatty acid. Now, odd chain fatty acids are fatty acids that contain an odd number of carbons. So for example, pentadecanoic acid is a 15 carbon fatty acid, and it's actually the most common odd chain fatty acid that's found naturally, and it's actually found in milk, interestingly enough. But I will say that odd chain fatty acids are fairly rare. They're a minority of the fatty acids that we eat. Even chain fatty acids are just much more common. Now, in terms of beta oxidation of these odd chain fatty acids, and I'm talking about saturated odd chain fatty acids, so no double bonds considered here. Beta oxidation actually occurs for these almost the exact same way as it does for even chain fatty acids. However, the final product will be a 3-carbon molecule called propionyl-CoA. And this propionyl-CoA is converted to succinyl-CoA via a 3-step process. And succinyl-CoA is an intermediate in the citric acid cycle. So this succinyl-CoA that's created from this propionyl-CoA can actually be fed into the citric acid cycle or the Krebs cycle to produce ATP. So again, for odd chain saturated fatty acids, they're broken down in beta oxidation, much like an unsaturated fatty acid. The only difference is that in the end, instead of ending with 2 acetyl-CoAs, you end with a single 3-carbon unit called the propionyl-CoA, which can be used to produce energy. I want to end this segment on beta oxidation with a few takeaways. Number 1, beta oxidation occurs in the mitochondrial matrix. And fatty acids have a special transport system that they use to actually get into the mitochondrial matrix called the carnate shuttle. Second, each beta oxidation cycle chops off 2 carbon units from a fatty acid. And then third and last, per gram, fats produce more energy than carbohydrates.
At Med School Coach, we know that getting into medical school is hard. In fact, 60% of pre-meds who apply to a medical school don't even get accepted. And if you want to get accepted into a top medical school, it's even harder. That's why more students than ever are turning to MCAT tutoring by Med School Coach. Our tutors are all 99th percentile scorers, have been through rigorous training, and can help raise your MCAT score by 12 points or more. You need every competitive advantage you can get to get into med school, so why not get one-on-one tutoring from Med School Coach? Our expert team will create a custom program just for you and help you master MCAT content where you need the help most. You'll raise your MCAT score. We guarantee it. Visit us online at medschoolcoach.com and use offer code PODCAST10 to save 10% up to $400 on a Med School Coach MCAT tutoring package. You can achieve your medical school dreams, and Med School Coach can help.
The next thing that I want to talk about here is kind of the opposite of beta oxidation, and that is fatty acid synthesis. Fatty acid synthesis occurs in the cytoplasm of a cell, and it occurs when acetyl-CoA begins to build up in the mitochondria. And this is basically the cell saying, okay, I have plenty of energy, right? I have all this acetyl-CoA that can be turned into ATP. I'm set on energy. Let me start actually building fat that then can be stored. And so this all begins by acetyl-CoA being transported out of the mitochondria by what's called the citrate shuttle. This is similar to the carnate shuttle I talked about earlier, only it helps acetyl-CoA exit the mitochondria. And basically what happens is acetyl-CoA reacts with oxaloacetate to make citrate, which diffuses out of the mitochondria. Then once the citrate is out of the mitochondria, citrate lyase in the cytoplasm converts citrate back to oxaloacetate and acetyl-CoA, and now you have acetyl-CoA free and floating around in the cytoplasm. Now, once you have this acetyl-CoA freed up and floating around the cytoplasm, fatty acid synthesis can begin. So in the first step of fatty acid synthesis, this acetyl-CoA is converted to malonyl-CoA. And at the most basic level, a carbon dioxide is added to the acetyl-CoA. So you're adding a carbon and 2 oxygen, and that makes it malonyl-CoA. At this point, fatty acid synthase, which is an enzyme, is like, oh, hell yeah, because it knows it's about to do its thing. And it takes malonyl-CoA and another acetyl-CoA and smashes them together. This creates a 4-carbon long chain. And then fatty acid synthase takes another acetyl-CoA and smashes that onto the chain, creating a 6-carbon chain. And this process just repeats itself until palmitic acid is created, which again is 16 carbons long. And basically, it's the reverse process of beta oxidation. You're combining acetyl-CoAs to produce a fatty acid. The only real difference here is that you need to start with a malonyl-CoA. I also want to mention that fatty acid synthesis requires energy. So it requires both ATP and NADPH. And overall, the process requires 8 acetyl-CoA, and again, those are what becomes the fatty acid, 7 ATP, and 14 NADPH. And again, that's NADPH, not NADH. Now, once these fatty acids are synthesized, they are added to glycerol to make triglycerides. And then these triglycerides are shipped to adipocytes, which are fat cells for storage. And one thing this might make you think about, or at least it makes me think about it because I'm interested in it, is the fact that fat tissue can accumulate without even eating fat. So if you think about it, glucose can be broken down into 2-acetyl-CoA molecules, right? And those 2-acetyl-CoA molecules don't necessarily get used to produce energy if you don't need it, right? So let's say that you have a huge excess of glucose, and that leads to a huge excess of acetyl-CoA molecules. What that's going to stimulate is the fatty acid synthesis pathway, and that's going to take those 2-acetyl-CoA molecules from the glucose and actually incorporate them in fatty acids, which are going to go on to be stored in fat cells that make up fat tissue. So you might be saying to yourself, well, duh, Sam, that's boring, but to me, that's super interesting. Your body can take carbohydrates and even proteins, as we'll get to later, and it can actually build fats out of those and store those fats for your later usage. Pretty sweet. To quickly summarize fatty acid synthesis, it's a process that occurs in the cytoplasm, and aside from the malonyl-CoA part, it's basically a reverse of beta oxidation. Before I move on to proteins, I also want to talk about ketone bodies. So, in addition to being used to produce fatty acids, acetyl-CoA can be used to produce ketone bodies. And there are really two ketone bodies that are useful in our bodies. That is acetoacetate and beta-hydroxybutyrate. And usually these are produced during periods of prolonged starvation, usually after gluconeogenesis has occurred. And in short, different cells in the body can use ketone bodies to produce energy. They're essentially converted back to acetyl-CoA and then used through the Krebs cycle to produce ATP. And I will note here that the brain actually is a pretty heavy user of ketone bodies because it does not use fatty acids for energy. So in short, ketone bodies are these small molecules that can be used as energy during periods of prolonged starvation, and they're also used by the brain when the brain cannot get enough glucose.
At Med School Coach, we know that studying for the MCAT exam can be challenging, especially for busy students on the go. That's why our team created MCAT Prep, the only all-encompassing study app built specifically for the MCAT. MCAT Prep by Med School Coach provides student access to extremely high-quality content and a personalized curriculum for free. The app has more than 250 videos, 1,000 flashcards, and 1,000 unique MCAT questions. Plus, MCAT Prep by Med School Coach allows students to create a personalized study schedule and track progress over time. You need every competitive advantage you can get to get into medical school, and now you can put the experts from Med School Coach into your pocket. It's the closest pre-med students can come to a personal tutor without spending a penny. Download MCAT Prep by Med School Coach for free at medschoolcoach.com/MCAT. Or download it directly from the Apple Store or Google Play Store. You can achieve your medical school dreams, and MCAT Prep by Med School Coach can help.
The next thing I want to talk about here is protein metabolism. I'll start again with catabolism or breakdown, and then I'll get into anabolism or synthesis. Protein and amino acid catabolism or breakdown begins in the digestive tract, specifically in the stomach. The liquid in the stomach called gastric juice has a very low pH, and that actually helps denature proteins. In addition, there is an enzyme in this gastric juice that is called pepsin. It's a protease and actually breaks down proteins into smaller oligopeptide fragments. After this occurs and some of the stomach juice goes into the small intestine, proteins are further broken down by pancreatic peptidases like trypsin, chymotrypsin, and carboxypeptidase. And these enzymes together cleave these oligopeptides into even smaller fragments, like mono, di, and tri-oligopeptides, as well as free amino acids. These very small peptide fragments and free amino acids are absorbed into the enterocytes in the small intestine, and then from there, they make their way into the bloodstream. From there, they're carried by the circulatory system to all the different cells around the body. Now, once these short peptide fragments make it into these different cells, they need to be broken down, and they're broken down into individual amino acids by proteasomes that are actually inside the cytoplasm of the cell. And from there, these free amino acids can go one of two ways. They can be further broken down, or they can be used to build more proteins. And since we're talking about protein catabolism or breakdown, let's talk about the first one. All right, so the first step in amino acid catabolism is what's called deamination. And this is the removal of the amino group from the amino acid. And according to the textbook Biochemistry 5th edition, quote, the alpha amino group of many amino acids is transferred to alpha ketoglutarate to form glutamate, which is then oxidatively deaminated to yield ammonium ion, which is NH4+, end quote. In short, the amino group of many amino acids is transferred first to alpha ketoglutarate to make glutamine, which is then deaminated to yield free ammonium ion. And now what you're probably wondering is what happens to this ammonium ion? This ammonium ion is eventually converted to urea, which gets excreted in the urine. And this all occurs via the urea cycle. And this whole thing kind of got me thinking, why is nitrogen bad? Why does the body need to get rid of nitrogen, right? Plants love nitrogen. When you feed plants, you give them soil that's got a shit ton of nitrogen in it. What's wrong with nitrogen in us? Why do we got to get rid of excess nitrogen? And this comes down to the fact that unlike carbohydrates and fats, proteins and hence amino acids are not stored in the body for later use, right? Muscles aren't like storage vessels for amino acids. They're usable muscles. They're not the same as adipose tissue or glycogen, which is the stored forms of fat and carbohydrates, respectively. Now, since you can't store amino acids, they would just build up until you had a really high concentration of them throughout the body. And at high concentrations, nitrogen is actually toxic. So the reason that our body needs to get rid of excess amino acids so badly is that number one, we can't store them. And number two, the nitrogen in them is toxic at high enough concentrations. Okay, now back to this process of amino acid breakdown. So as I said, the first step is deamination. That gets rid of the nitrogen. Now, after that, the remaining part of the amino acid, which is just basically the carboxylic acid with some R group, is converted into products that feed into gluconeogenesis, ketosis, or both. And it's important to know which amino acids feed into gluconeogenesis, called glucogenic amino acids, and those which feed into ketosis, or the production of ketone bodies. And these amino acids are called ketogenic amino acids. The L amino acids only feed into the ketogenesis pathway. That's lysine and leucine. On the other hand, tryptophan, threonine, tyrosine, isoleucine, and phenylalanine can feed into both glucogenesis and ketogenesis. And you can remember these using the mnemonic triple T intellectual property, or TTTIP, which sounds like some sort of fancy patent firm. Again, that's triple T intellectual property. And you just got to remember that the P stands for phenylalanine. And now all the other amino acids that are not lysine, leucine, tryptophan, threonine, tyrosine, isoleucine, or phenylalanine feed into the gluconeogenesis pathway only. Therefore, they are only glucogenic amino acids. And there are some really cool graphics that show kind of where each amino acid feeds into the Krebs cycle or the gluconeogenesis pathway. It's pretty cool to see. You could just Google glucogenic versus ketogenic amino acids. I'll put a link in the show notes if I can. And that stuff's not necessarily going to be on the MCAT, but it's very interesting just to kind of see which points in this whole process the amino acids feed into. The last thing I'll mention here is that amino acids can also feed into other biosynthetic pathways. For instance, serotonin is synthesized from tryptophan. The nucleotides are synthesized from aspartate, glycine, and glutamate. And there's other biosynthetic pathways that these amino acids feed into, but I just want to give you a little taste of what that looks like. So overall, proteins are broken down into amino acids. Then these amino acids are deaminated. And then the remaining bits of these amino acids feed into gluconeogenesis to produce carbohydrates, or they feed into fatty acid ketone body synthesis to produce fatty acids and ketone bodies. Hence, even proteins can be turned into fat.
Med School Coach, the company that sponsors this podcast, is doing a merch giveaway. So all you got to do is go to merch.medschoolcoach.com and use the promo code podcast5 for $5 free at the merch store. And this is literally $5 free. It's not like $5 off 20. It's not 5% off. It's just $5 free. And there's like pre-med stickers, mugs, t-shirts, A bunch of good stuff there. So again, if you want $5 to spend at the Med School Coach merch store, go to merch.medschoolcoach.com and use the promo code podcast5.
All right, the last little bit of stuff I want to talk about here is on protein anabolism or protein synthesis. So when proteins are broken down into individual amino acids and they make their way into cells, these amino acids can be either broken down further or used to build new proteins. And I'll talk about that second one now, which is building new proteins. So cells in the body are pretty much constantly producing proteins. According to Portman's et al. In a paper titled Protein Turnover, Amino Acid Requirements, and Recommendations for Athletes in Active Populations, the body synthesizes 3 grams per kilogram per day of proteins, meaning that in a single day, I produce something like 240 grams of protein. That's like half a pound of protein a day. That's crazy. Now, the process of protein synthesis should be very familiar to you. This is just transcription and translation, right? This is just the central dogma of molecular biology. And so I'm not really going to review that for like the 50 millionth time that you've seen that in your life. But what I do want to mention that I haven't mentioned before and what you should know for the MCAT, and that's how amino acids we get from our diet are incorporated into proteins that the cell builds. And there's one step in particular I want to point out. So free amino acids need to be conjugated to their correct tRNA or transfer RNA in order for them to be eventually incorporated into a protein. In this reaction, the conjugation of a free amino acid to its correct tRNA is catalyzed by an enzyme called aminoacyl tRNA synthetase. And these enzymes are pretty damn cool. They are not only able to attach amino acids to their correct tRNA, they can also check the work of other aminoacyl tRNA synthetases. They can essentially come up to the tRNA that's conjugated to an amino acid, inspect it, and kind of check it out. And then they can hydrolyze the bond between the tRNA and the amino acid if they discover that the wrong amino acid has been added to the wrong tRNA. And according to Wikipedia, the accuracy of the aminoacyl tRNA synthetase is so high that it's often paired with the word superspecificity. Pretty badass. Now, once an amino acid is added on to a tRNA, that tRNA is considered to be charged and can be used by a ribosome for protein synthesis. So overall, proteins can be absorbed and broken down into individual free amino acids. These amino acids can either be 1, broken down and used to produce glucose, fats, or ketone bodies, 2, used to make another biomolecule like the neurotransmitter serotonin, or 3, used to build more polypeptides or proteins. So that's it for the Fat and Protein Metabolism podcast. As always, if you found this helpful, go leave us a review on iTunes. It helps us in the rankings, and I always love seeing it. If you hear any kind of error, feel free to email me, and I'll hopefully fix that. If you want to just tell us that you like the podcast too, I always love hearing that. Thanks again for listening, and hope to see you next time.
Thanks for listening to Prospective Doctor's MCAT podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on the MCAT or getting into med school, check out medschoolcoach.com for the experts.